Hi, I'm Gina Boat with 3D Molecular Designs. Let's go over some tips for assembling and disassembling the nucleotides in the dynamic DNA kit. The 12 base pair set of the dynamic DNA comes with 24 nucleotides that students can assemble. Each nucleotide consists of one of 4 nitrogen bases, an adenine, a thymine, a guanine, and a cytosine. There should be a sugar group and also a phosphate group. To assemble a nucleotide, we will start with the base adenine, and we're going to insert the blue post on the swivel portion into the sugar where there is a space for it. Next, we'll attach the phosphate group. Look for the thicker post with the paddle on it, and then we are going to line that up with the slot in the phosphate. And you have an assembled adenine nucleotide. You can do the same with the other bases and assemble them so that you have all 4 bases for base pairing. Next, I'll show you how to disassemble one of these. The trick here is to hold at the base and pull firmly straight out. Likewise, between the sugar and the phosphate, hold at the base and pull straight out. Thanks for watching. Find more assembly instructions at dynamicdnakit.com.